So we're going to be talking about RNA here, and how information is contained in RNA that can be read into protein. So in that sense, we're talking about information almost in the same way that we're talking about reading the pages of a book. That is, this makes sense because when you read the changes of the book, our brains translate that into meaning, and when you read ribosomal RNA sequence, you need something to translate that into a meaningful thing for proteins synthesis. And it turns out this is a big, so here is my message, okay? And I'm going to read this for you. It's a wonderful message. It's a message in English, but I want to give this lecture in Japan, so if there's anybody could help me make this. Okay, it was the best of times, it was the worst of times. It was the age of wisdom, it was the age of foolishness, it was the epoch of belief, it was the epoch of incredulity, it was the season of light, it was the season of darkness, it was the spring of hope, it was the winter of despair. We had everything before us, we had nothing before us. We were all going direct to heaven, we were all going direct the other way. Yes, well, it's a Tale of Two Cities, but this is the opening lines of A Tale of Two Cities by Charles Dickens. It's one of the great opening things. So it's beautiful, and there are no mistakes in this. That is, we can all get some meaning from this. But if you were to look at the initial transcripts from eukaryotic genomes, this message would look sort of like that. Okay, with huge amounts of nonsense. There are parts of it that seem to be. It was the best of times, it was the worst of the times. It had mad noting, blah, blah. So, I mean, you throw up your hands, or maybe if you were a cryptologist, you would sit there and think about it. But what has happened is that we need the great translator and that will clear this up, and then that is the spliceosome. So the spliceosome is going to clear up this thing, and we will get the real message, and that's the subject that I want to talk about. That is, I'm telling you that in all of eukaryotic cells, the messengers, the initial transcripts from the thing are almost always interrupted with nonsense by introns. And sometimes, so this would have been one of these nonsense parts right here, and those have to be removed by the spliceosome in order to make sense, and it has to be done correctly to the nucleotide in order for this truly to work. And Jim mentioned this, but there could be alternate things happen in which instead of splicing from here to here and here to here, we would splice from here over to here, and we would make two different proteins from the same. It's called alternative splicing, but I'm not going to talk about that in this lecture. Okay, so here's what you see when you look at the human genome, and we know what the human genome sequence is. Of course, we may know that the hundreds of thousands of them. Do you hear this? That you can now sequence a human genome for $1,000. Okay, so, and at the time when I started, you couldn't sequence anything. Okay, so the average links. So these are the introns. But the square boxes are the exons. The exons are small, around 145 nucleotides, but they can be very large. The intron's length in humans is 3,000 nucleotides or so, but they can be huge, 500 kilobases. So how in the world, in that sea of RNA, can you correctly make the excision to the nucleotide? It's a big problem And so could be an average in the human genome 8.8 exons per gene, but it can be as high as 145. So there's a huge amount of splicing that needs to be done in order to make these messenger RNAs. And so it has to be done with high accuracy, otherwise you'll never have anything that works. And so here are the signals, and they're meager. You can best see in yeast the length of the letter here corresponds to the frequency of which you see a nucleotide. This is at the 5' splice site, so the gene coming here, and then this part has to be. So it's the GTA, TGT, but this is not nearly so conserved in human genomes. And then there is a branch point sequence. We'll see about that in a second. The TAC, TAAC, almost always present as that sequence in yeast. It's virtually hard to find when you look at the human genomes. And finding there's a small sequence here at the 3' end splice site, CAG, that's usually there too in humans. Okay, so the mechanism of splicing, this was strange, okay? Nobody expected this, and when they got the first results, everybody was very puzzled. But it turns out that what happens is that you do this by two metal catalyzed reactions, which are common chemistry in most enzymes that have to do with nucleic acid metabolism. And so there's a 2' hydroxyl, because this is RNA, at this branch point A, and that's the nucleophile. It attacks this phosphate, and then you get OH here, and all of this is done by magnesium that are coordinating this phosphate. We're going to see this in a lot more detail in this lecture. And so this magnesium is stabilizing the nucleophile, and this magnesium is stabilizing the leaving group. And then in the second step, it's the end of the 5' exon, that is the nucleophile, and the 3' splice site, which is the leaving group. So it's a two transesterified reactions, and these reactions can occur at close to equilibrium, so that there are the same number of phosphate bonds found as there were at the beginning. They're just rearranged. So all of this happened in the 70s, but a lot of it happened fairly quickly, actually. That some really central puzzles came about. First of all, there were these transposons, primarily in bacteria, but in some organelles as well, that were in the great discovery of, as Mike Yaros will tell you, I think of one of the great discoveries of our era is the discovery that RNA could act as an enzyme. And these RNAs can be called self-splicing because they have the enzymatic activity to splice themselves. And the one that Yaros discovered used the nucleophile as a free 3' hydroxyl of guanosine. This does the reaction. The group 2, as I told you, uses as the nucleophile an A in the TAC sequence. That's the messenger RNA. But to our surprise and excitement, another kind of group of self-splicing RNA, the group 2 RNAs, work by precisely the same mechanism as the pre-messenger RNA. But then I and my group and other people spent 15 years looking at tRNA, and since we did this first, we thought we might be the first ones to find a mechanism of splicing, but unfortunately it's not the messenger RNA splicing doesn't use the same, so it's quite beautiful, but it's entirely conventional chemistry. There's no self-splicing going on in tRNA splicing. And all tRNAs do not have introns. Some of them do, some of them don't. Why any of this happens, I'm not going to talk about that. Okay. So, but here's really the central problem that we need to worry about is that here there's the sort of folded structure of a simple group 2 RNA. And so it has catalytic activity and it has a conserved secondary structure. I should say that I'm going to talk a lot about this loop D5. It's a part of the catalytic center of this group 2 RNA, but this is a typical pre-messenger RNA sequence, and you can look around in this distance, which could be 2,000 nucleotides between the 5' splice site and the branch point, and there's no conserved sequence, and there may be some functional sequences there, or functions that do something else, but so there's no structure, no conserved structure, and as I show you in the consensus sequence, minimal conserved sequence. But if you were to make this splice wrong, then it gets the wrong protein. So it's got to be done right. So this is what actually does this. Is a huge RNA protein for pre-messenger RNA. It catalyzes pre-messenger RNA. It has 5 snRNAs as SNRPs. These are RNAs that carry out the functions of splicing, both the folding of the substrate so that it can act as a substrate, and also serving, as I'm going to argue, as the catalyst of the reaction. But there's more than 100 proteins, and for many of those we know and have studied, and we can talk about the functions of them. I'm going to talk in particular in my second lecture about one very important protein in the spliceosome. But it's very, very complicated. But in the end, because this works in the same mechanism as group 2 splicing, we conclude the simplest hypothesis is that it's RNA catalyzed. And I think I'm going to be able to prove to you in the 2 lectures I give that that's really true. But why all the proteins? And the self-splicing group 2 goes with no ATP requirement. What's the ATP doing in splicing? Okay, well, this is a stepwise. The spliceosome doesn't exist as a machine like that can bind to the intron and correctly splice it. On every event, the spliceosome is assembled via several steps that we now understand fairly well. It's the same in humans as in yeast. Yeast is the one that we study. And so one of these things, U1 SNRP recognizes a 5' splice site, U2 recognizes the branch point, then a trio of SNRPs, U4, U5, U6 join. There we have 5 SNRPs together at this point. And then the mysterious and interesting thing is that it gets rid of 2 of those, U1 and U4, to make the active spliceosome. And then it carries out the reaction, and then this has to be a cycle in which you then it releases the messenger RNA and goes back again and reforms. So here's the guiding hypothesis. There's been, since the days that 20 years ago or so, that was realized that the mechanism of splicing, pre-messenger RNA splices, is the same as in group 2. So unlike most introns and sRNAs are conserved in eukaryotic evolution, that is, we can't convince ourselves that there's an RNA structure within the intron itself that's involved in splicing, but because they're quite conserved in evolution, the sRNAs could have. And in each spliceosome assembly event, the sRNAs must interact with the pre-messenger RNA. This is, of course, the assumption, to form a catalytic active site similar to that seen in group 2A. And some experiments have now just been done, which I find quite convincingly prove that statement, or at least make it highly likely. Okay, well, now this is the complicated side, and I don't think I'm going to go through this all. These are the secondary structures of U2 RNA in 2 different forms. I'll talk about that later. The U5 that's as part of the U4, U6 RNA. And so one thing I'll do in order to just show you the crucial rearrangements that occur in the RNA molecules as a spliceosome assembly occurs is that U4 is a strongly base paired. The melting temperature of that is about 50 degrees centigrade. It's quite stable. And I told you in splicing assembly, U4 leaves, and U6 forms this extended internal stem loop with a bulge here, and this loop turns out to be an important part of the catalytic center of the spliceosome. It's the most conserved of the uRNAs. And I might just say that this is a really nice slide from Fabrizio and Lerman, that there's an incredible flux of proteins going on as well, although we're not going to talk very much about this, but you would need to really know this, you need to know why it's happening. So the 44 proteins to add with a triple SNRP, up to 35 proteins are discarded. 9 proteins are added, 2 are discarded, and so forth. So this is really a dynamic thing, and what are all these proteins coming and going for? So it's quite interesting. I think it's going to keep people busy for a long time. It's complicated enough, interesting enough, but I think we sort of understand. That means that there'll almost certainly be surprises. Okay, well, so then there's the really interesting thing that why ATP? So there are ATPases that are required for every step in assembly, and then including the discard pathway. And these are so-called dead box helicases. In some cases, they have been shown to have helicase activity, RNA-dependent helicase activity. In other cases, it can be shown that they are involved in the removal of proteins that are bound to RNA, so that the exchange factors. And I'm going to say quite a bit about PRP16, which is occurs is a dead box helicase that's involved in the second step of splicing. So if you make a mutant in PRP16, you accumulate the lariat intron and the 5' exon intermediates, and you don't finish splicing. Okay, so now this is profound. Okay, so I was talking about the fact that with all this small amounts of information, how can you get the accuracy that you need? So this is the proposal that John Hopfield, a physicist, he was a colleague of mine at Caltech. And Jacques Nino, I also knew him. He's French. He was at the Salk Institute at the time I was in San Diego. These are very smart for this time. But they reasoned that if in biological reactions where the specificity is needed, that you could get extra accuracy by having an off-pathway step that required ATP hydrolysis in order to make it, in order to drive it towards the active step. So, and furthermore, they clearly said that if the failure to reject an incorrect substrate in one step is a probability P, you can get much greater accuracy by doing many steps. Then you get to the accuracy of P to the nth. And since P is a fraction, you get greater and greater, less and less probability that you'll make a mistake. Okay, so there you are. I mean, I just showed you we've got 8 steps, and so we should get P to the 8th accuracy somehow here. Okay, so now I'm going to show you exactly how all of these ideas started, and this is really with Christine Guthrie. And I have to tell you that I no longer have a life. I have a lifetime, but I have no lab. I work in Christine Guthrie's lab. She's my wife, and she did these experiments in her lab some time ago. In any case, she's a yeast geneticist, and they set out with a branch point mutant that you're supposed to have an A there, and they put a C. And they isolated suppressors of this. PRP16-1 was one of the first isolations of this gene, and it was able to use C in the branch point reaction. And so that meant that it made PRP16 more error prone, and that was how we were able to translate that. So the idea is that when you made an active site in the ATPase, you slow down this rate of discard and allow incorrect products to go through into the product, and that's of course an error-prone reaction. And then she went on to show that if you making many mutants in the active site of PRP16, that you would get these. Okay. So now I'm going to go on to talk about the active site of the group 2 RNA. And one of the thrilling things that really has happened in the last 4 or 5 years is that we have a high resolution structure of this. This happened in Anna Marie Pyle's lab at Yale, and a very good postdoc tour found one intron that was small and did not require a protein for activity, and they made 100 variants of this slicing off loops, and they found one that crystallized to 3.5 angstroms, and so they've solved that structure. It's an active group 2 RNA, and here is a representation in the helices of this. And I told you that the interesting thing for the active site is this D5, and that's actually enclosed by helices. It's right in the middle of the enzyme. And so then she also got crystals of the first step reaction. So you can see how, so let's look at the reactive site itself. So it has, looking closer at it, it's formed, it has a series of triple base things that are resulting of the bases from this part of the ribozyme from J 2/3 in here, and those form 2 metal binding sites. But in addition to that, this was in a much higher resolution structure, they found 2 potassium ions. So potassium is required for the in vitro systems that we use, and this is being used in a very interesting way here. Yeah, okay, I think yes, this is a better slide. Okay, so so here are the 2 magnesium ions that are poised to interact with the sessile phosphate in order to affect the first step. And those are catalyzed by many interactions, both with side chains of amino acid and with the bound potassium ions. So then she did an experiment in which she crystallized this enzyme in sodium. So sodium won't support catalysis in group 2 enzyme. And what happens is that just by the rotation of 2 bases, this 377 and I'm reading this word. 2 bases rotate out away from the active site, and it completely abolishes the 2 metal binding site. Now, here's the most important thing, okay? She did experiments then to show that this is an obligatory intermediate in going between the first and the second step of splicing. That is, you have a step that binds metals here and can carry out the catalysis. You must go through a step which is not catalytic in order to go to the last step, in which you now bind. Unfortunately, they have not a crystal structure of the second step, but you can. It's very likely that that's quite similar to the first step, because the chemistry is the same. Okay, so here's a cartoon of this. In the first step, we have potassium ions and metal ions bound, and then we go through these 2 base rotations, and we have no metals bound. The silent step. Then we go to reach the second step. We can again bind metals and carry out the second step of splicing. So really, I'm going to talk a lot about this silent step. Okay, and the reason why is that if this is an obligatory step for the group 2 ribozyme, it must be that it's an obligatory step for the spliceosome. Now I'm going to talk about a recent paper from 2 brilliant people at labs at Chicago, John Staley and Joseph Piccirilli. So Piccirilli is an organic chemist. Staley, one of the major people in the splicing field. He was a postdoc of Christine's, and he's done beautiful work in the last 10 years at Chicago. We can leave this field, but it's in fine hands. It's no problem. Okay. So, I need to say a couple of important things here. Actually, some 20 years in my lab, we figured out how to reconstitute active yeast extracts with a synthetic U6 RNA by inactivating the U6 that's in the extract, allowing it to be degraded, and then adding a synthetic U6. This is a U6 RNA that's been synthesized entirely chemically. That's a pretty big feat, but it's now can be done. And that restores activity, and that means that you can make changes to the U6 substrate that you have made in order to understand the various biochemical questions about its function. So they made some 20 or 30 U6 variants that have a pro-S or a pro-R ox. So the phosphate bond is chiral. So it makes a difference whether you put the S in the pro position or the R position. In many cases, they made both. This is not trivial because in the synthesis, you get a mixture. So you have to separate the oligonucleotides that are pro from those that are R. The pro-S from those are pro-R. And when you get an S here and it's involved in either structure or it doesn't interact. Sulfur does not interact with magnesium well, but heavier, softer metals like cadmium and manganese does. And so this is a so-called metal rescue experiment. I'm going to talk more about this rescue in a minute. Okay. So, first of all, now we're just looking at the second step, and so this is the aberrant mobility on a acrylamide gel of the lariat intron exon 2 intermediate. Here's the released area, and so if you have counts here, it means you went through the reaction. So they looked at 20 of these, and it turns out that many of them that are in the center of U6 will rescue only in the presence of cadmium. So those 5 residues are important, and they are boxed here. They were known to be important sequences because you can't make mutants in them that work well in U6. And Patrizia Fabrizio and I had done experiments 20 years which we substitute, not chirally separated, but the chiral phosphorides into these things at specific places, and these are the positions that we found were important. So 5 of these things are involved in either the stability or the catalysis of catalytic action of U6. Okay, so now comes the point where you separate the men from the boys. So, oh, sorry, yeah, yeah. I think I just told you this story. 5 of them turned out to be active. Okay, so now, rescue experiments proved that certain non-bridging oxygens are required, but they couldn't prove that they're required for catalysis. They could have a structural role. So these guys did even more complicated chemistry, in which they made the 5' splice site with an S in the bridging position and one other S in one of the chiral positions. And to prove that these are involved in catalysis, they wanted to see whether you would get interaction with these that would be synergetic, and I'm going to explain this in just a second. These are very, very difficult to splice. In any case, they will only splice in the presence of cadmium, not manganese. So. Okay, so but for several of these, particularly U80, there's a synergy between having a particular pro-R sulfur in the U6 and having the sulfur at the 5' splice site. This is for the first reaction. They did this experiment for the second reaction too, but I'm just not going to go through that because it's the same kind of experiment and similar sorts of results. I'm not going to explain more about why this experiment works. So magnesium can form intersphere contacts with ligands such as oxygen, but resist intersphere coordination with soft ligands such as sulfur. So So it's inner sphere contacts, but the softer metals, such as magnesium or cadmium, readily accept and sometimes prefer sulfur as an inner sphere ligand. So you can get these strong the cooperative interactions between, say, an sp phosphorus here and an sp oxygen there, and that's the reason why it's a combination of substrates and U6. It's in one position can lead to synergy. Okay, and so you see this in the case of, for the first step of G78, U80 in both of its forms. So I'm going to conclude this complicated slide, and perhaps you need to pay attention to the second step, because I didn't explain those experiments, but the take-home lesson is the same. That what you see as ligands in the domain 5 has a strong resemblance of the group 2. It's a strong resemblance to the U6 internal stem loop, and very similar ligands are active, and so these two things prove that the active site is very like the RNA, and that's the title of the and furthermore, and this is going to be important for our next talk, you can actually now make a plausible three-dimensional structure of RNA in the spliceosome, and making this triple. Here it is in the group 2 spliceosome. Here it is. Sarah, a graduate student, or a postdoc in our lab, made this slide, and it's a very plausible one involving all these crucial things, and you can make the same kind of triple structure. And here's a diagram that shows the kinds of triple structure bases that are formed in this very similar either D5 or the U6 internal stem loop. So the conclusions at this point, and these conclusions figure heavily in the arguments that I will make in the next slide, because these are important points that I think one couldn't have really said before. That is that the active site of the spliceosome is nearly identical to that of the group 2 ribozyme. This suggests strongly what we had believed, and I'll show you more evidence for this in the next talk, that they have a common evolutionary origin. That is that a group 2 transposon, the splicing originated from a group 2 transposon. And lastly, the first step is nearly identical to the second step active site, another important point. Okay, so I will give my detailed acknowledgments at the end of the second talk.